Okay, today we'll discuss what lac operon is. Okay, in order to do so, we'll go back to a cell. A cell has its DNA inside the nucleus, and the DNA contains the codes for the formation of the baby. And the work of the DNA is not done, it's not over. Just because the formation of the baby is over, the work is not complete. It still needs to keep the living organism alive. In order to do so, it has to produce thousands of types of enzymes required every hour by living organisms. So all this information to produce these enzymes is written on the DNA. So the DNA is constantly being copied. And the organism needs to know which part of the DNA, since DNA is a very long strand, it needs to know which part needs to be copied and when. It cannot copy all the DNA, all the segments of DNA all the time. So as and when required, it needs to copy segments of it. Okay? So this monitoring of the copying of the DNA, or the exact or the particular type of segments of the DNA, is known as gene regulation. Gene regulation is nothing but the regulation of the copying of DNA segments as and when required. Okay? Now, lac operon is a very sophisticated, you can say, or a very fancy way of regulating this copying of genes. It has been studied in the prokaryotes, and we take the example of E. coli or Escherichia coli. Okay, this is an enteric bacteria, meaning it's in the intestine. So what this bacteria eats is the food that we eat. The food, not that fat boy from the hostel. This food that we eat contains milk sometimes. This milk contains about 2 to 8% of lactose. And lactose, as we know, is actually made up of, it's a disaccharide, meaning it contains two sugar molecules called the glucose and galactose. Now, what the bacteria actually wants to do is, it just wants to break the glucose and lactose in order to obtain glucose, so that it can use that glucose for glycolysis. Glycolysis is, the breakdown of glucose in order to release energy. With this energy, it can perform its works. Now, the bacteria has a set of codes known as lac Z, lac Y, and lac A. These are the genes that produces an enzyme. This enzyme can break lactose into glucose and galactose. This galactose is used for the process of, during the process of glycolysis, it can give out energy. So the work of, the objective of the bacteria is to gain energy. And in order to gain that, it has to produce these enzymes which can break down lactose. So now when lactose is present, it activates, or it begins to produce these enzymes. Let's say enzymes are produced. So these enzymes will break down lactose, and glucose is obtained, and from that glucose it gets its energy. So its objective is fulfilled. But when lactose is not present, when lactose is absent, because humans are not taking in milk all the time. Our food does not contain milk all the time. So those are the times where the bacteria does not want to produce the enzyme, because that would be a big waste just to produce huge amounts of these enzymes which break down lactose when there's no lactose at all. So that is not what it wants to do. So lac operon is this process of regulating the production of enzymes that break lactose. Right.